Let's talk about control of the cell cycle and what happens when that control goes unregulated. So there are various different types of cells in our body. Some of them continuously divide, like the cells on the surface of your skin, cells covering the internal coverings of your intestines. Some of your cells, they do not grow. After they mature, they start dividing. So when it comes to the cell cycle, those kind of cells that stop dividing, mature cardiac muscle cells, for example, or nerve cells, after they finish their final maturation into what they're supposed to be, they exit the cell cycle and go into a phase called the G0 phase. There are also cells that temporarily go into a G0 phase, so it's not a one-way journey. For cells like this, mature cardiac muscle cells and nerve cells, once they exit, they don't go back in. But a lot of cells in our body exit the cell cycle when there's not a need for them to divide anymore, and when they receive a signal, they go back into the cell cycle. So there are signals that are given to cells, and they tell them, okay, it's time to get back and start dividing. Now the question is, okay, how does the cell know when it has entered a new phase? And how does the cell does quality control? Well, the process of cell division has evolved over millions and millions of evolutionary time, and in addition to becoming really prepared before the cell division happens, the cell also has quality control positions within the cell cycle, which are called the checkpoints. And there are 3 of them. The one checkpoint at the G1 phase, which is right before the cell enters the S phase. The second one is at the G2 phase, towards the end of the G2 phase. And the last one is right in the middle of metaphase, which we call the M checkpoint. So I have created a border for each of these of a different color so that we can start talking about them one by one. At the checkpoint in the G1 phase, the decision is the cell has to make a decision whether or not the cell needs to go ahead and divide and whether or not it should go ahead and synthesize more of its DNA. And that's usually a place where the cell checks for damage to its genetic material. If there is damage to the genetic material, there's really no point making more of it. So this checkpoint is a place where the cell checks that, and there are molecules that are involved in making that decision. The checkpoint at the G2 phase asks the question as to whether the DNA replication is complete and accurate, and whether or not the cell is ready to go and divide. And then the mitotic phase, there is a checkpoint, and it checks to see whether sister chromatids are correctly attached to the spindle fibers, and so whether or not the cell can proceed. So now the question is, what are the molecules that are involved? The major molecules that are involved in determining when it is that a cell is ready to go into a different phase in the cell cycle, when is it time to go. One of the major regulators are this pair of molecules called cyclin, and it's kind of fitting the name is cycling because it's involved in the cell cycle. And then CDK, or cyclin dependent kinase. So what does the kinase mean? A kinase is a protein that adds phosphate functional groups to other proteins. So if you remember, we talked about a phosphate functional group, and we usually show it like this, but it in actuality has a lot of oxygens attached to it. It's a very negatively charged functional group. So A kinase is a molecule that adds negatively charged these phosphate functional groups into other proteins. Now, these cyclin-dependent kinases are available throughout the cell cycle at a constant level, but the cyclin molecules, their levels fluctuate. So it goes up and down depending on which phase of the cycle the cell is in. Now, this cyclin-dependent kinase isn't really active unless its partner cyclin binds to it. So when the cyclin levels are low, like places like here and here, this kinase cannot add any phosphate to other proteins. It's inactive. It is only when there's enough cyclin produced and bound to this CDK, it is then that the kinase can start doing its work. So the fact that you have a cell cycle and there are different phases into it, these phases are determined to a large part by when there's enough of these cyclins accumulated. Now, there are different types of cyclins. Their names are listed here, and each of them accumulates to a turning point, for example, their maximum point at a particular phase of the cell cycle, and each of them have their own partner cyclin-dependent kinase. So the cyclin B pairs up with CDK1, cyclin E, CDK2, so on and so forth, and each of them have a function at a particular phase of the cell cycle. So here's a graph showing the concentration of each type of cyclin and different phases of the cell cycle. So you can see that cyclin D kind of goes to its maximum around the end of the G2 phase. Cyclin E becomes maximum at the transition between G1 and S phase. Cyclin A gets to its maximum level towards the end of the G2 phase, and cyclin B gets its maximum towards the beginning of the mitotic phase, which we said it works at the metaphase stage. So as soon as these high levels of cyclins are accumulated, then you have enough cyclin CDK that you can trigger a change in the cell cycle. That's kind of the checkpoints where you can signal to the cell whether or not the next phase can move forward. So a little bit more about cyclin CDK. So the CDK, cyclin-dependent kinase, it's an enzyme. And if you remember from our discussions with enzymes, we said they are allosteric regulators, either activators or inhibitors of enzymes. Well, this is an enzyme that's an allosteric activator. So when this activator is at a very low level, remember we said the levels go up and down. So this activator at the very low level, CDK, is inactive, that kinase. But when there's enough of it, then the cyclin can bind to the CDK, and then another protein comes and adds a phosphate to this enzyme. If this cyclin isn't there, enough of it isn't there, this phosphate isn't added. So now the cyclin CDK is active. Now this enzyme can add a phosphate to another protein and push it towards moving to the next stage. So this would be a target protein that is normally sitting in an inactive stage. It's waiting for a signal to, let's say, pass through the G1 into the S phase or pass through the metaphase stage. So as soon as this CDK phosphorylates this protein gets activated, and then the cell goes past through the cycle. So one of the examples of how this works is this protein called retinoblastoma protein. This retinoblastoma protein is a protein that normally stops the cell from moving past the G1 phase into the S phase. So it's a negative regulator of the cell cycle. Now, normally, the retinoblastoma protein is bound to another protein and is inhibiting it. This protein turns on genes that allow the cells to start growing. So again, in the absence of active CDK and in the absence of enough cyclin, this retinoblastoma protein binds to this molecule and stops the cell from passing into the G1 phase. But when cyclin CDK are active, then they come and phosphorylate this retinoblastoma protein, which no longer can bind to this protein. And now cell cycle can proceed. So that's an example of how CDKs work. So now this is a really nice coordinated way of controlling when the cell divides, when it's not supposed to divide, but things can go wrong, mutation can happen, and cells can become out of control. There are times when no matter how many checkpoints you have, cells can ignore those checkpoints and just rapidly and without limitation, they keep growing and they give rise to cancer cells. So this is supposed to show a tumor cell. These blue ones are the cells that no longer can be controlled. Those cell cycle controls are out of whack. So the cell keeps growing and generates a tumor. So how is cell growth, the extent of cell growth regulated? So right now, before these few slides, we looked at molecules that are involved in deciding when it is time to switch between one phase to another. Those are checkpoints. But there are also proteins that are involved in either pushing the cell towards growth or stopping the cell from growing when it's not under good conditions to grow. So there are two classes of genes that control cell growth. One class is tumor suppressor genes. And that retinoblastoma protein that you saw was one of them. Tumor suppressor genes, normally their normal function is to block cell growth. The other class are proto-oncogenes. These are proteins that normally promote cell growth. So their normal job is that they push the cells towards growth, but their activity is controlled. Now, what happens when they go out of control? Sometimes mutations destroy the structure of tumor suppressor genes. And when you do that, when you inactivate tumor suppressor genes, then there is no mechanism to stop the cell from growing. Okay, so the cell growth goes out of control. In another instances, with respect to proto-oncogenes, something happens that takes that control away that would keep these in line, these proteins in line, so they keep dividing. And therefore, the cell goes out of control. Now let's take a look at a couple of examples of how these things can happen that can give rise to cancer. So let's take a look at a particular tumor suppressor gene, and one that is called the guardian of the genome, because it plays a major role in making sure that if the cells have damaged DNA, they do not go through the cell cycle. It stops them. So if the DNA of a cell is damaged, or the cell has abnormalities, or there's limited lack of oxygen, this p53 protein does two things. If the damage is not too high, the DNA damage is not too high, the p53 protein, which is a tumor suppressor, it arrests the cell cycle and gives the cell enough time to repair the damaged DNA. And then once the damaged DNA is repaired, it allows it to go through the cell cycle. Now, another thing that p53 does is that sometimes it pushes the cell to kill itself. And this happens when the DNA damage is really so high that there's no really hope for the cell to have a normal cell cycle. In a way, it prevents cells from becoming cancerous. And this happens when you go into the sun and get a lot of sun on your skin, let's say if you don't wear sunscreen, and then the cells on your skin get their DNA gets damaged by the UV lights rays of the sun, and the DNA of your cells on your skin gets so damaged that p53 just pushes those cells towards committing suicide. That's why your skin peels when you get a sunburn. That's the work of p53. So p53 protects our genome, but what happens when p53 doesn't work, even though the cell has DNA damage and there are conditions where the cell is in a state that it may have abnormalities and become cancerous? Well, p53 can't do any of these things that it did in here. The cell cycle continues, mutations accumulate, and the cell can become cancerous. Now, we talked about proto-oncogenes, and we mentioned normally are involved in promoting cell growth. But mutations can lead to proto-oncogenes to become cancerous. They can turn from becoming a proto-oncogene to becoming an oncogene. An oncogene is the bad form of a proto-oncogene. So how is some of this stuff, what are these mechanisms? So sometimes genes change their positions. Genes can hop from one place to another, or sometimes cells can have really unstable chromosomes, and the chromosomes can break and rejoin and cause a gene to change its position. And that change in position causes the gene, the normal growth stimulating protein, to produce in large amounts. So when you produce a large amount of something that normally promotes growth, well, the more of it you have, the more growth it's going to promote. Another way of proto-oncogenes to become oncogenes is when gene duplication happens. And gene duplication does happen very often, and sometimes when genes are genetic contents of a cell is not stable, sometimes it makes extra copies of itself, and that leads to, again, accumulation of excess amount of this gene that normally pushes the cell to grow. And the more you have, the more growth you're going to have. Finally, you can have mutations in a gene that normally supports the growth, which either it produces more of the gene product or makes the gene product to stay longer. So again, you're going to have an excess of the protein, or you can mutate the gene so that the protein is hyperactive. It cannot no longer be controlled, and no matter what signals the cell receives, this protein keeps pushing the cell to grow. So these are all the different ways that a proto-oncogene can turn bad and start pushing the cell towards too much growth when it's not necessary.